Oh, thank you very much, Catherine, for a very nice introduction. So today I'm going to talk about the role of velvet antenae in maintaining healthy brain aging. I have to say this is a very small pilot study. So the research was conducted at Otago University because the results are quite preliminary. We haven't really published yet. So it will be really good if you just hear but do not, make it public at this stage. Right. So aging, everyone cannot avoid aging, which is not a very nice process. So in general, aging is a process which happens, in both human beings and also animals. Aging is also a major risk factor for a number of neurodegenerative disorders. For example, Alzheimer's disease and Parkinson's disease, and those two affect our health enormously. So this kind of age-related cognitive decline actually is related to some brain regions. They don't really function well. For example, one area is the prefrontal cortex. The other one is the hippocampus, which is a part of the temporal lobe structure. So in the brain, as everyone knows, we have lots of neurons. So those neurons are the fundamental unit for our brain and also for the nervous system. Okay, I just see the layout is slightly different. So basically, those neurons need a very good blood supply, to maintain their normal function. So the neurons need to coupled with the blood vessel supply, and to ensure that brain region has adequate and also accurate blood supply, okay? And because the neurons need good oxygen and also glucose supply. So the neurons in the blood vessel, we need to have some mechanism to control the cerebral blood flow, which is involved one enzyme. And you can see from the blood vessel, we call it endothelial cells. So one enzyme called endothelial nitric oxide synthase, in the short term is eNOS, can produce a gaseous molecule called nitric oxide. So NO derived from eNOS plays a critical role in terms of maintaining normal cerebral blood flow. So in the brain, we have this kind of quite complex structure, okay, we call it neurovascular unit. Because some neurons are probably are quiet, but other neurons are very active. When the neurons are activated, of course, we need very nice supply of oxygen and also glucose. So from this diagram, you can see we have a neuron here, but a neuron needs to talk to the blood vessels in this way through all those different types of cells. And some cells looks like the star-shaped, we call it astrocytes. So those neurovascular units plays a very, very important role in terms of control cerebral blood flow and decide how much blood needs to be given to a specific regions. So as I said before, neurons needs to be coupled with the blood vessels in the brain, and really depending on the neuronal activity in that area. So this phenomenon we called neurovascular coupling. So in rats and mice, as you can see here, because they have whiskers, all right? But the whiskers needs to project to a certain brain area, and one region called barocortex. If we stimulate the barocortex, and we can see the activated neurons in the barocortex. If we stimulate whiskers, we can see increased neuron activity in the barocortex, and also accompanied with increased cerebral blood supply in that particular region. So this is basically a commonly used experimental approach which allows us to assess the neurovascular coupling integrity in certain brain region. I have to say this phenomenon, normal function, optimal neurovascular coupling integrity is really critical to maintain our normal brain function. So it has been very well documented in the Alzheimer's disease and also in advanced aged brains. The neurovascular coupling function is significantly impaired. So the deer velvet antler has been used for over 1,000 years in traditional Chinese medicine. So in the market, I think anti-aging effects and also memory-enhancing effects has been advertised or widely accepted. However, to the best of my knowledge, so this kind of effect have not been systematically assessed, okay, in both human beings and also experimental animals. In 2017, the Velvet Antler Research in New Zealand called a proposal, so aiming to test the hypothesis and whether repeated small doses of velvet could improve cerebral microcirculation in middle-aged and also aged animals. So we came up with a research proposal by doing a pilot study just to focusing on the cerebral vascular function. So this slide shows a general experimental design of our research. So as you can see, we used male Sprague Dawley rats at age of 12 months. So for rats, this is a kind of a middle-aged rats. You know, if we convert this rat age to human age, looks like probably 30 to 40 years old of human beings. So after a couple of days habituation in the experimental environment, we started treating those animals with New Zealand freeze-dried velvet antler powder. So what we did, we basically make this powder in the water suspension, and we treat animals at a daily dose of 1,000 milligrams per kg, which is a very high dose for rats. So the total duration of treatment was 19 days. After 2 weeks of pre-treatment, then we put animals in the behavioral testing. We test animals in several, behavioral apparatus to assess different aspects of learning and memory. So after the treatment, we then, on the final day, we performed a pericam experiment. This involves a real-time microcirculation imager. Basically, where wild animals stay alive, and we can look at the regional cerebral blood flow under baseline condition. And we also carried out a study to look at neurovascular coupling by stimulating rats in the whiskers, and we can detect the corresponding area, the blood flow changes, basically. So at the end, we count animals, we collect brain tissue, and to assess the cerebral vascular markers, a number of markers. So the whole experiment was conducted by my team. It involves Dr. Hu Zhang and Mrs. Nicola Coley, Dr. Yu Jing, and also a PhD student, Ashwani Hariharan. So the whole study was conducted in a blind fashion, so they don't really know the treatment group, okay, keep everything blind. Only me know which group, which animals in which group. So let's have a look at the results, what kind of results we got so far. So we weighed animals every day since animals were delivered to our university, okay, in the lab. So as you can see from this diagram, by the way, the black square indicates the velvet group, okay, means the rats in the velvet treated group, and the open circle is the control one. Looks like in the velvet treatment for over 19 days didn't really affect animals' body weight. I guess this is something good, okay, so indicates no adverse effect, okay, to animals. So then we tested animals in the open field apparatus. This apparatus is very simple, just an open field box, okay, with the top is open. We put animals there for 5 minutes and just let animals freely moving around for that duration. Then we can, imaging animals, we can analyze data offline. So when we analyze the distance traveled during the whole 5 minutes period, as you can see, the velvet animals, they were quite active. They walked a lot, okay, during that 5 minutes. So we also quantified one typical behavior. This is called rearing behavior, because rats are quite curious. They were so interested in the novel things. If you put animals in the novel environment, they would put a front paws and looking around. You can quantify the duration of this behavior. So again, as you can see from this lower part of the diagram, so the velvet group reared more. So this behavior is really critical because reflex exploratory activity. So this ability is really important for animals to navigate in the environment, okay, and required for the spatial learning and the memory. So the conclusion here, we just said, well, although it's not statistically significant between the two groups, but it looks like the velvet-treated animals are more active. We then train animals in the Morris water maze task. This task is really simple, just like a quite big swimming pool. And we put, a platform, because rats don't really like to be in the water. They want to struggle and get out. And so they find out, wow, there's a platform, I can escape. They just go straight to the platform and also use the external visual cues. That's why the water maze is a commonly used spatial learning and memory test to assess the ability. So on the first day of the test, we initially just put animals to swim for 1 minute, okay? So it looks like, in terms of the swimming ability, there's no difference at all between the two groups, which is identical. Then we introduced a platform in the pool, but animal could see. Okay, there's a visible platform called cued navigation, so there's no difference between the two groups. Then we put a platform hidden, so animals couldn't see. So both groups generate longer path lengths to reach the platform. So by the way, the path length, if the longer means poor performance, shorter path length means better performance. It's very interestingly, for the second trial of that hidden platform, once the velvet animals knows, oh, I went there before, and the second trial, we released animals from a different starting point. They basically swam directly to the platform, really suggesting those animals have better performance, okay, for this particular task. So for the next 3 days, we make the task even difficult, okay? So we basically get animals to learn 2 platform location within a day, but with 30 or 180 seconds delay between the sample phase and the test phase. They really had to work very hard to remember what's going on during the sample phase, therefore can guide their performance during the test phase. So we didn't really see significant effect, but looks like, the velvet-treated animals, they generated shorter path lengths at both 30 seconds and 180 seconds delay intervals. So again, they performed better, okay, in this particular water maze test. So on the final day of the treatment, we used real-time microcirculation imager to look at the basal level of cerebral blood flow in the brain. So this is the results. So this is the baseline perfusion, okay, presented in the y-axis. We have a control and a velvet. So no difference between the two groups. Remember, they only had 19 days treatment. So we then use this whisk stimulation paradigm to look at, how the barrel cortex in that brain region, how the blood flow changes during the stimulation. So we can see the velvet-treated animals, looks like, they displayed greater perfusion response. So which means when you stimulate the whiskers, the neurons in the barrel cortex will have to working hard, okay, immediately because the sensory information gets to there. And this activity would have to couple with the blood flow, because just like when you run, you need to have lots of energy, probably eat more to support your hard work, right? So at the end of the PERICAM study, we collect the hippocampus to look at a number of markers. So as I mentioned before, in the blood vessel, there's an enzyme called endocellular nitric oxide synthase, and eNOS-derived nitric oxide plays a very important role in terms of maintaining normal cerebral blood flow. So the hippocampus is a very large structure in the rat brain, and it contains 3 subregions. We named CA1, CA3, and the dentate gyrus. And each subregion has different function in terms of their role in the learning and memory processing. So not many researchers will be able to dissect the subregions, but this is the kind of routine work in my lab. So we simply separate CA1, CA2, 3, and also dentate gyrus, and we do the Western blot to check the eNOS protein level in each subregion of the hippocampus. So what you can see here, this is eNOS protein. We converted, expressed as percentage of controls. So in the CA1, C3 brain region, there's no significant difference between the 2 groups. But interestingly, in the dentate gyrus, and we can see this is significant increase in eNOS protein expression in the hippocampal dentate gyrus. So we further look at the blood vessel density across the 3 subregions of the hippocampus by using this antibody called the RECCE1. So the top 2 panels illustrates the immunoreactivity profile from the control animal, and the lower 2 labeled B, and shows the data from a typical velvet treated rat. So you may agree with me, it looks like the lower part has more the wiggly blood vessels, small blood vessels, okay. When you carried out a quantification, looks like it is a small but a significant increase in terms of RECCE1 immunoreactivity in the brain region in the velvet-treated group, okay? So if I put just all those data together, just to summarize, I have to say this is a pilot study. We used male SD rats at age of 12 months, and those animals treated with New Zealand freeze-dried velvet antler supplementation. We didn't really see any change in the body weight, but we found those velvet-treated animals appears to be more active in the open field test. In the water maze task, they also appeared to perform better. Okay, and one task, it was significant, a difference between the 2 groups. Although the neurovascular coupling study didn't really show significant difference, but I guess you agree with me, the velvet-treated animals have greater response to whisker stimulation. And we also have Western blotting evidence and also immunohistochemistry to show the eNOS, protein levels increased in DG and have increased RECCE immunoreactivity in the hippocampal CA1 region. So those preliminary findings, I think, support the beneficial effects of velvet antler supplementation in the middle-aged rats, particularly for the male SD rats. However, I have to say there are a number of issues we need to consider. So first of all, this is really just an animal study, okay? And the experiment was conducted under this very particular, very special experimental design. And the sample size is quite small, and the velvet treatment, the dose is very high. And the treatment period is also quite short, only 2 weeks. And then we tested the behavioral performance, and we assessed cerebral blood flow changes, on the day of 19 days treatment. So I guess the results are quite interesting. Of course, we would like to carry on more research to provide more evidence to support this the claim, velvet would be beneficial, probably can enhance our brain aging. Okay. Finally, I just want to thank Catherine and for the support from the Vance. Thank you very much for your attention.
Thank you very much, Dr. Liu. So, as Dr. Liu said, this is just a taster preliminary study. Vance will be reporting this study in a suitable fashion to those in our supply chain. If anyone's particularly interested in taking this further or wants to know more, then please do get in contact with us. Now, Jamie's indicated that we might have 2 minutes for questions. So if anybody's got a question for either Jeff Asher or Ping Liu here, please raise your hand and we'll get a mic to you.
Yeah, just to put it into some context, I was just wondering if that dose rate, if you compared it to a normal velvet capsule for human consumption, what sort of dose rate is that, or how many velvet tablets would you be taking a day?
I think probably you can. You can answer.
The precise answer is considerably more in a human, yeah.
It's very high dose.
I know you showed us the mean, and it was a small study, but was there any difference in the variation between the two groups?
I think in general, the variation, as you can see, the data are presented. I think it's pretty similar, but for some difference, you can see a clear trend, but this is not statistically significant. I haven't got time to do the power test. I think certainly we need to increase sample size in order to get significant results. Yeah, and another thing, because we used 12 months old animals, so if you test animals in the behavioral task by comparing 12 months versus 4 months, you can see some subtle difference. Okay, you can see some behavioral changes. But if we compare 22 or 24 months old animals with 4, the behavioral impairment just so dramatic. So I guess if we repeat study by using aged animals, we probably will get more, far more clear pattern. Yes.
Could you use female rats? Would it be any different?
I think probably female rats will benefit probably even more from this treatment because we can see in general female rats perform even poor as compared to the males at the same age. Yes.
Thank you, Dr. Liu. Any more? There's a question at the front here, and Dr. Asher's available if anyone has a question for him.
Given the test was only over 14 days treatment, it's not very often we'd be given something and saying, take this for 14 days and you'll be sweet. So presumably just even prolonging the treatment period, is going to have some impact. What's your thought on what that impact would be?
Yeah, I think prolonged treatment probably will increase the benefit effects for sure. Yeah, so some study really depending on the funding anyways. I would like to do the long-term treatment study to see how those animals, performed.
Do you think that the improvement of the treated rats was due to improved blood flow only?
Well, I guess the improvement, we can see clear behavioral benefit from the velvet treatment. So this could be due to multiple factors. Because the Vance provide this small pilot funding, so we could only do much. We have already collected the blood samples and also different brain regions. We can do more work to look at, the underlying mechanism which support this beneficial effect, although it's quite preliminary results.